Welcome to the third session. I will be very brief about my relationship to David, which was extremely nice and scientifically productive. It generated one paper some 9 years ago, and it was, as you know, David is everywhere. So this particular one was on the analysis of protein sequences using an algorithm that David devised that has to do with looking at short peptide content within longer sequences. And we were analyzing the evolution of olfactory receptors, sense of smell receptors, which have been on my mind for many, many years. And we got some very interesting insights. A shared student, Asaf Gottlieb, did the work. He's now in Texas, David tells me. And he was even affected by the flood, which is something that no decent Texan can afford not to happen to him or her. So our second session begins with Professor Eytan Ruppin, whom I know for many years. He received his PhD in computer science from the Tel Aviv University in 1993. He was formerly a professor at the Tel Aviv University in the School of Computer Science and Medicine, and now he's professor at the Department of Computer Science at the University of Maryland. He's also director of the Center for Bioinformatics and Computational Biology there. And he told me that he's actually, am I supposed to say that? Eytan, where are you? Can I? That he's moving to the National Cancer Institute. I was doing a lot of bioinformatics interactions with the National Cancer Institute, and I've always noticed that they need a guiding hand to do things right. And Eytan is moving there to do just that. Eytan is an expert in computational systems biology. He studies metabolic systems modeling of different organisms. In one of these organisms, humans, about which he will speak today, he focuses on metabolic alterations in cancer and aging, and these studies are aimed, hopefully, to identify novel drug targets. The title of Eytan's talk is, importantly, Can We Defeat Cancer? Eytan, please.
So you hear me like that, or I need to put something? Okay, sure. Okay, is there a laser pointer somewhere? No? Speakers beforehand? Yeah, I can. And my talk is somewhere? Which? Oh, thank you so much. Okay, and I'll do that. Thank you. So, right. So first of all, thanks a lot for the kind invitation. And Mazal Tov to David, of course. It's really a déjà vu for me to be here. You know, I've spent 20 years of my life in Tel Aviv, and it's an amazing place. This hall has traumatic memories for me because I studied medicine here before my PhD, and most of the exams were in this hall, and I always didn't know anything. I was very busy copying. So from everyone I could, you know, I would sit there. And right, so whatever, I managed to graduate, yeah, you know. Yes, so let's begin, and feel free to stop me anytime, okay? It's a rather challenging talk. I never gave such a talk. The last public talk I gave in Tel Aviv resulted in me meeting my wife and marrying her. So that was a quite rewarding event. I have no expectations in this field now. But let's see. Perhaps dinner in the evening would be right. So we'll see. How do I? Yes. So There's no pointer. I need a pointer. It's a matter of confidence. Nobody has a pointer. Oh, thank you, Doron. Great, great. Okay, never mind. If anyone has. Anyway, so 25 years ago, I met David. I was in my PhD, doing my PhD in Tel Aviv. I was working with Isako Mellerson, who is here, with Marius Usher, who is here, and with David. And we worked on Alzheimer's disease, building neural network models of attractor neural networks, and trying to understand why there is a sharp memory decline in Alzheimer's disease and relating that to phase transition in neural networks. In short, we had lots of fun. Okay? I will talk and will aim to describe to you what's the current happenings in cancer, which is really, truly, thank you so much. Thanks a lot, Doron. Which is truly ongoing a transformative period now, as I hope I'll manage to convey to you. And it's not a hype, you'll see, and I'll aim to describe. I should say that I'm not a clinician, so I'll give you a basic science perspective. And the task, this talk is rather daunting. Let's see how it goes. So first of all, I will begin with a short introduction to what is cancer, what's currently the state of the art. Then I'll talk about precision-based medicine. Each of you who opens Ynet, like I do every day, you can see on the left side of the first page of Ynet, there's always these semi-ad articles about foundation medicine and getting precision medicine stuff in cancer. I don't know if you see these articles. So I'll explain what's going on, and then I'll move to the main focus of my talk, which is cancer immunotherapy, explain what it is, ve'emania, and finally some closing remarks. That was supposed to move forward. Yes. So, as you know, by and large, tumors can be benign and tumors can be malignant, that is cancerous. And when they are malignant, you usually broadly categorize them according to the tissue of origin. As part of the developmental of the human embryo, there are three main tissues of origin, and we, for various reasons, both clinical and deeper biological, we distinguish the cancers into epithelial ones, mesenchymal ones, and hematopoietic blood-related ones. And this is just to give you, one picture is worth 1,000 words. So these are two counts of growth. One is benign, and you can see, so these are colon polyps. On the left side, this is how in an endoscopy a benign colon polyp looks. So you can see it's pretty well organized, demarcated, stuff like that. This is the histological picture. This is a microscopic slice of the tissue, and you can see that this is the lumen. This is, one layer, the first layer of the colon, cut like in a cross-section. This is the muscle tissue. And this is how the tumor looks. This is how it looks microscopically, and this is how a cancerous tumor in the colon, a cancerous polyps look like. You can see that the tissue is in total disarray. You can sense the population of evolving cancer cells that penetrate, metastasize, and disrupt the whole normal tissue organization. So, the distinction is pretty clear. And in the last 10, 15 years, there every 3 years appear, updates on a series of papers, which is very famous in cancer research, which is called the hallmarks of cancer, by Hanahan and Bob Weinberg. And cancer is not just about unlimited growth or penetration. So there's quite a few phenotypes that occur. So it becomes self-sufficient. It doesn't need growth signals to grow, in contrast to our normal cells. It is insensitive to anti-growth signals. It evades certain programs of cell death by which we can regulate and shut down normally overgrowth in the body. It has limitless reproductive potential. These cells, in contrast to healthy cells, can divide forever. It secretes different small molecules that create their own blood supply into the tumor. It invades, it metastasizes, and it's characterized on the genomic level by a tremendous amount of genomic instability, mutations, changes in the genome, and which support the evolution, sometimes the rapid evolution of these phenotypes, these hallmarks of cancer. And I just pulled up the recent progress report of the AACR committee in the last meeting, just to tell you that I will talk about new and exciting stuff that is now happening in the front of cancer. However, progress is being made, and very important progress is being made even in the absence of that, by the slow grounding improvements of the methods we have. So just to give you two numbers in the earnest of time, there is about a 25% decrease in the last 15 years or so in the number of cell deaths per 100,000 population in adult cancers, and there is about 35% decrease in the rate of cancer deaths per a certain amount of the population in children's cancers, okay? So, and you can see that the slopes are pretty consistent, so progress is even being made, but I want to say that I expect, and I hope to convince you, that due to these new breakthroughs that are occurring just in the last 3 years or so, the slope will become much higher if one can predict, and if I'm right. And this just to show you that's the same thing. This is in adults. You can see this decrease in cancer mortality in the population. And in parallel, you can see that there is almost a doubling in the amount of cancer survivors that live with us, among us today. And one of the challenging issues now is actually taking care of cancer survivors that has to ensure, try to prevent as much as we can that cancer will not reoccur, et cetera, et cetera. How do I go back? I don't know, beyond me. I'm a professor of computer science, but whenever there's... What? Thank you. I call my wife or ask Ranit. It doesn't work. It does? Yes. I'm just stupid. I want to show you this slide. It's part of this report. I think it's rather telling and important. So you can see the survival rate, okay, the 5-year survival rate, how many people survive after 5 years in case of an initial diagnosis, for patients where the disease, they were lucky and the disease were caught at very early stages, versus patients with the same disease, but which was only diagnosed in advanced stages. So you can see the huge difference in the 5-year survival as a matter of early detection, the time where we catch this disorder. So I will talk today about these recent exciting developments that are occurring in our ability to treat advanced disease. But if only we had a system for early detection, even with the current treatments, we could do so much better. And I'm not even talking about prevention, but if we'll have time, I'll talk about some issues happening there. So at the end of my talk, if I have time, I'll come back to recent exciting developments in early detection. Okay, but just think. If we could only detect it very early, it will actually make a lot of our current efforts to treat advanced disease redundant, right? So we'll get back to that. So just a short comment on that. Holy Grail. How many of you have heard of a company called Grail? Right, so Noam has heard about it. It's a small startup in the Silicon Valley that has raised $1.2 billion. Okay, as a startup because they have they claim they have an amazing technology to do early detection of cancer very early, and we'll come back to that. So another, and the last slide in these intro slides is this one, just to show you again from this AACR recent progress report. So they have collated, so, thank you so much. תודה רבה. וזה גם מעביר את הסליידים? זה רק ענק כזה במקום הדבר הזה? לא, תודה. So they have collated the risk factors. So there has been in the last 2-3 years some very exciting but controversial papers that claim that most of the risk of cancer is genetic. Okay, and if we can have time, I can explain. But certain amount of cancer is, of course, environmental or things that we can do. So this just shows you the percentage contribution out of the environmental contribution factors. Okay, you with me, guys? Yeah? So you can see that tobacco is about 30% or so. Right? What happens if I press this button? I hope not a nuclear war. Nothing happens. Oh, here. So about 30% or something are tobacco, about 20% are obesity, about 15% are pathogens, H. pylori, as you know, some of you, whatever, that may cause gastric problems and stuff, is a major pathogen, and it's a famous Nobel Prize in medicine, another heroic story, and other things. So you can see this relative contribution of these factors to cancer. Good. Now, cancer treatment, after we discover cancer, is based on 5 pillars. 3 of them are the classical ones: surgery, radiotherapy, and cytotoxic chemotherapy. And the 2 ones which have become more recently into the field are precision-based therapy and immunotherapy, on which I will focus. However, I want to say a few words on chemotherapy. Chemotherapy is based on the idea that we give drugs that basically, to put it very simply, kill proliferating cells. And the idea is that cancer cells are more proliferating than other cells in our body, and hence you get a selective killing of the cancer cells, but a lot of side effects because many of our healthy tissues also proliferate, and then you get also a lot of side effects and stuff like that. And the idea is to develop treatments which only selectively hit cancer, and to develop treatments that actually can bring cure. Okay, so that's the basic setup. And until quite recently, cancer has been mainly characterized by the histology, how it looks under the microscope, the origin of site, the level of spread, clinical, phenotypic markers, and mainly was treated by these 3 basic treatment modalities, and also the supportive curves, we'll come back to that, have been quite limited. While now we are treating cancer based on a very detailed molecular characterization, we have much more treatment modalities, and in quite a few of the cancers you will see we are doing much, much better. So a few words still on chemotherapy, just to say, you have seen this decline, 25% in adults, 35% in kids, from 1990 until now. This has been mainly still due to improvements in chemotherapy and treatment in general. And I just won't go into the details. I'll just say that constantly, all the time, physicians, scientists are working very hard to improve protocols of chemotherapy. It's based on combinations, clinical trials, how to do it, what regimens, how to carefully dosage, adding adjuvant chemotherapy for different kinds of cancer, finding biomarkers. This is highly important. We'll come back to that. Which subpopulations will respond best to which protocols of chemotherapy? Learning that many times we were too aggressive in the chemotherapy regimens, and actually that we can do much better with lower doses in some cases, and using lower dosage will actually then improve the long-term mortality of those who survive. Because with chemotherapy, for reasons we cannot discuss, you can hopefully many times cure, but then you get recurrence after time, and it's related to the aggressiveness of the treatment. So if we can learn how to achieve the cure with lower dosage, we then increase on the longer run the survival of the survivors. Okay? So a lot, with all the excitement about the new therapies, I don't want to undermine what has been achieved with current therapies. And I want to say that I will talk mainly today just on the treatment, but there is huge and important other fronts where a lot of research and advancement is being made. If I can talk about it later, about prevention, understanding the basic biology, making the quality of the life of the treatments much better than before. We have much more possibilities, and as I said, better treatment for those who survive. Okay, so after setting the stage, I want to go and talk a bit about precision oncology and then talk about chemotherapy. Before I do that, is there any questions up until now? Is everything okay? Good. Yes, please. Yes, yes. Oh, absolutely. You're absolutely right. Thank you. So there's huge differences in the treatment, survival prognosis in different cancers. In the interest of time, I'm just, aiming to give a very global picture. You're absolutely right. Yeah. I've shown one picture with you of different cancer types where I compared the survival in early stages and late stages, and you could appreciate it that the different cancers, these prognoses are very different. Absolutely. Okay, so the first new real development that has, of those that I want to talk today, that has occurred, is been occurring, I would say, I don't know, 10, 15 years, but is accelerating is precision oncology. So as you know, in 2001, there was a revolutionary event in biology, right? And that was what? Guys, what happened in 2001? The human genome, right? The race between the private company, Celera, and the public NIH, right? A dramatic human event. Converged in parallel publications in Nature and Science in the same time and whatnot, heroic stories. They published the first draft of the human genome. By the way, just to show you how little we know, when they published the human genome, how many genes were thought to be in the human genome? 100,000. How many are they today? What? Well, so protein coding genes is 19,600 now and shrinking. Okay? Let me tell you another shocking thing. What percentage of our DNA are coding genes? It's about 2%. Okay? So a lot of the DNA is doing highly important functional other stuff. And finally, of all our genes, 15 years after the Human Genome Project, how many of the genes do we know their annotation? Do we know what they do? Do we know their function? What percentage? About 50% we think we know what they are doing. However, the Human Genome Project was awesome, unbelievable. It cost only $3 billion. There has never been a bigger buck for the bang for the buck, or buck for the bang, whatever, than the Human Genome Project. It's amazing. And it enabled in cancer research to start sequencing cancers, tumors from patients, and looking on the annotated genes and identifying mutations in genes. So first we learned that some subset of our genes, let's say we have 20,000, about 300 of them are what we call cancer driver genes. Okay, they are divided to oncogenes, genes where a mutation activates a gene in a way that promotes cancer, and tumor suppressor, genes where a mutation deactivates a gene in a way that promotes cancer. So there is a very long tail of mutations that occurs in the genome, but in most of our genes, they're quite very infrequently, but like in 300, 400 genes, some of them are outlaid here across different cancers, you can see very high frequent mutation rates in these cancer driver genes in specific cancer types. So finding these mutations laid the possibility and started the race of developing drugs that will aim to target the genes, these genes when they are mutated, when they are found in a given patient, and thus selectively and specifically attack the cancer of a patient according to the mutations found in these cancer driver genes. Okay? Very simple idea, right? Of course. Drug development is complicated and whatever, and you need to identify the mutations, and you need to identify them in the patients, you need to attack them, and it's a great idea, but there are problems. We'll come to that. Okay. So in 2005, the National Cancer Institute started an amazing project, the Cancer Genome Atlas, which by now has about 10,000 samples, tumors of patients of 30 different cancer types, where we collected information on their DNA sequencing, on their gene expression, different kinds of proteomics, metabolomics, methylation, different kinds of biological information. And everybody, the Ishtor, are harvesting, mining this data in order to understand what's going on and looking for drug targets and cancer-specific vulnerabilities. And if a patient has a tumor, these days we can take the tumor, we can sequence it, we can analyze it on the different layers and try to give precision-based drugs in according to the specific tumor. So this has revolutionized the way we look about. So we don't talk anymore about lung cancer or colon cancer or even according to some histological types, but actually we view the cancer types within, here in this case it's non-small cell lung carcinoma, within the specific characteristic mutations in these cancer driver genes that characterize this cancer. So we actually view these cancers of a given histological type as collections of a variety of cancers of molecular subtypes, each which can be treated and targeted specifically in accordance to these vulnerabilities. And there was a lot of hype, which is always regrettable, about the rise of targeted therapy. This is probably, 10 years ago, and just you can see the titles for yourself. This is just another picture that is worth 1,000 words. This is a few years ago already, but you can see it. So this is the membrane of the cell, and cells represent certain molecules that are receptors, and these receptors respond to different extracellular events, and they, via other protein-protein interactions, which are called signaling pathways, they convey information to the nucleus of the cells, which alter the expression of the genes, and that's how cells operate and communicate and react to the environment. So all these kinds of mutation-based specific drugs target different molecules within the cell machinery in order to try and restore and revert the malignant behavior of the cells according to the specific mutations and alterations that occur and return them back to the healthy state. And you can see how different pathways are attacked. But you can already get a glimpse of the problem that these are very rich pathways with many alternative routes in them. So when we attack specific routes, what may happen, what may likely happen, that these cells will find alternative routes in order to express their oncogenic, carcinogenic potential, and resistance is quite frequently likely to evolve. So you would then think that perhaps we would attack it with a combination that hits the cell in different targets in a precise manner, but then in what problem would you go into? That these combinations tend also to be harmful to healthy cells. And the whole idea initially is to find selective targets that selectively kill cancer cells. So you can see the complexity of the challenge. This is actually a pretty amazing slide. So on the top, this is a time scale. This is just for lung cancer. On the top, you can see different mutations that have been described by basic scientists that occur by mining the tumors in patients, that occur in a sizable fraction of the patients, and hence there is an impetus to develop drugs, okay? So these are the mutations that have been discovered, and these have been the drugs, the precision-based drugs that have been developed by the pharma in response to these molecular mutations that are discovered. You can see a matching between them to capitalize on these mutations. And you can see the number of the drugs that appearing to personalize treat. And those of you who read, these stories in the media have seen perhaps stories about patients who are already quite a few years living with aggressive lung cancer, which before was a death sentence, basically. And what happens? It's the race of the race of the drugs against their cancers. So every 1 or 2 years, whatever, their doctors find that they have some vulnerabilities that can be attacked by one of these drugs. They get the drug that keeps the tumors in check. They may even disappear, but they are not completely cured, because as I've shown you, this alternative pathways resistance is quite likely to emerge. But then if they are lucky, new drugs appear that may attack some vulnerabilities that they have, and some patients have already been lucky enough to go 3-4 cycles, and remedies are kept, and and who knows? Right? So that's a situation, and a rising rapid of molecular informed development of precision drugs, but resistance too frequently emerges because of the complexity of the system. So I think I've said all that. So precision medicine is great, but it currently looks only on about the status of 300 cancer driver genes, and there is a need for alternative approaches that look actually on the whole cancer genome, on the state of the whole 20,000 genes. Why just the 300, 400? So my lab is among the labs in the world which are working on trying to develop precision-based medicine based on the whole genome, okay? But I will not have time to talk about that. If you want, you can ask me. People are looking on combination therapies, a lot of clinical trials. When I'm talking a lot of clinical trials, I want to give you the number. In the NCI major hospital alone, there are hundreds of clinical trials going on precision-based medicine, new drugs, and combinations. In the world, I would assume now that there are thousands. Okay? So there's a lot of activity. But the main issue is how to scaling laws, which, as we build these combinations, would, in the language of physicists here, which would scale better? Hopefully the selective benefit to the treatments or the magnitude of adverse side effects and mortality and morbidity that are associated with that. Cool. So now we have reached the main topic of the talk. And David, how much time do I have? 10 minutes. I asked David. Okay. Let's go. So, I want to describe to you some truly exciting stuff that has really come to the clinic only in the last 3, 4, 5 years. And it's the first time which on a large scale, not just on individual tumors, we are seeing a vision, a window of opportunity for curing patients. Not this race of achieving temporary cures and resistant emergence and so on, but a real shot. So what's the story? So for a long time, of course, we know that the system can recognize and react against cancers because it's a foreign body, it's mutating, it's expressing what we call neoantigens. Antigens, the things, the proteins on the membrane which the immune system recognizes, okay, that are different from healthy antigens. So we know that. But we also know that cancer is amazingly smart and tricky in evolving mechanisms for evading the immune response. So it's a race, okay? And this population evolves much faster than our immune system, okay? Furthermore, different immune systems promote cancer growth. And finally, the immune system, I don't have the time, it's an amazing system. It's an amazing complex system of interactions with different subpopulations which we are making important strides in understanding, and this understanding has led to our ability now to tackle the tumor, but it took a very long time. Okay, it's a heroic struggle of 20 years, of those of you who have read the stories about Kite Pharma and stuff like that, that only very recently has come to fruition. And one of the world pioneers on that is a guy called Steven Rosenberg, who heads the cancer immunotherapy in the NCI, an amazing person who, like, for 25 years is working on that. And for 20 years, basically, he was mocked. Okay, so if you're talking about persistence in science, that's an amazing example. So this, to set it up, this is the tumor, and these are the immune cells. Okay, the active guys that are relevant in killing cancer are called T cells. T cells come in a large variety of population, helper cells, killer cells, CD4, CD8, whatever, a complex picture that we need to understand. Now, the lymphocytes come to the tumor, so they need to reach the tumor. They need to penetrate it, okay? And what we want to do for them to do is to kill the cancer. But the cancer secretes cytokines, small molecules that inhibit the activity of these tumor T cells and so on. And furthermore, to complicate the issue, the activity of these T cells, which kill the cancer cells, is dependent on another kind of immune cell, which is called a dendritic cell, which is an antigen-presenting cell, which mediates the molecular events from the cancer, the antigens, to the T cells and is responsible for their activation. And they communicate with each other by secreting different molecular cytokines. So this is the system that we want to study and characterize. And I want to briefly describe 3 kinds of immunotherapies. So just to set the stage, this is these dendritic cells. They take antigens and they present them to the T cells, to the tumor cells. Then they attack the tumor, and so on and so on. And I want to talk about cancer vaccines. I will talk about adoptive cellular therapy, and finally about checkpoint blockade. And I want to explain to you intuitively what is the story in each of these approaches and what is the current situation. Okay, so the first one is cancer vaccination. The idea is pretty simple, right, guys? I mean, cancer is a foreign body. It presents different antigens. We have a fantastic history in medicine in developing vaccines against foreign bodies, right? Against what? Bacteria, viruses, cholera. Yeah. Why not take this approach and develop vaccines for cancer? It's so simple. So people, of course, have been working and trying that for 20 years. And the idea is to take tumor cells, break them down in the lab, take the proteins, the membrane proteins, whatever, prepare vaccinations, inject them back to the body, and get vaccinations. In the sense, vaccinations, I mean not preventive, that's another dream, but just to excite the T cells, the immune cells that kill the cancers, to excite them to grow, to do clonal selection for the specific T cells that kill the specific tumor in the patient. Okay? So this is a wonderful idea, but it doesn't work. Doesn't work, regrettably yet, for reasons which are not completely understood, but what works quite a bit with mild success, for example, in prostate cancer, is taking out the dendritic cells, these antigen-presenting cells from the patient, taking them outside ex vivo in the lab, loading them up with antigens from the tumor's patient, putting them back to the patient, and this has moderate success, okay? So this simple approach is nice and interesting, but only partially and moderately works. As many times is the case. What is simple doesn't work so well. That's life. Next is a more serious issue, adoptive cancer therapy. So here, what we are doing is we are taking out the T cells from the patient. Okay? We are taking out the T cells themselves. Okay? We are stimulating them via versus specific molecules because they are exhausted. The tumor secretes stuff that dampens the activity of these T cells. We take them out either from the lymph nodes or from the tumors. We reactivate them. Okay, we reactivate them by exposing them to specific antigens for the tumors from the patient's tumor. And we get these tumor-specific excited T cells. Then we inject them back into the patient after depressing his the rest of his immune system. That exacerbates the positive effect. And this can kill cancers. This can kill cancers. And it has shown a considerable benefit in melanoma, but now we have better treatments. The problem is that in solid tumors, this doesn't work. You do the user, but the T cells don't reach the tumor source. They're not effective there for reasons which we yet don't understand. The idea is great. It's partially working. It's just in very recent years, but still it works only on some tumors or not on the majority of solid tumors. So it remains to be seen. The next possibility is CAR therapy. This is what you heard about, right, with the recent Kite Pharma and other companies, Novartis, whatever. This is what Steven Rosenberg and others have done. So we take it to the next step. We not only take the T cells out, but we actually do sophisticated molecular engineering, and we change the receptors of the T cells. We engineer them to attack specific neoantigens on the tumors. So it's sophisticated molecular engineering combined with all the molecular biology and the ways we learn to enhance the activity of T cells in adoptive cell therapy. Guys, are you with me? I hope it's not too fast. Is anyone still with me? Yes. Thank you. Izako. Tzaddik echad. Wonderful. So the issue with that it's pretty amazing, but you need a major neoantigen which is expressed in a very large fraction of the cancer. So for example, in ALL, okay, in acute lymphocytic leukemia, we are fortunate that there is a certain antigen called, never mind the details, which is expressed in almost all of the cells. So we know that, and we know the guy, and we know how to engineer the T cells specifically to attack him. And we can do that, and that is a very effective treatment when it works. This is just a picture that I took in my phone. I was Thursday, Friday in an amazing workshop in NCI with all the biggest guys who made this field happen. It was really exciting. So just like me and the Japanese postdocs were taking photos. And you can see, so this is the time in mouse. This is with relapsed ALL after conventional treatments. You can see the cumulative survival. It's dismal, it's dismal. And with CAR therapy, you can see that in 70% of the patients treated, they actually get, this is still ongoing, but for 2 years, there is no further decline. Those that have responded well, they seem, hopefully, carefully, to be cured.
Time is up.
Seriously. Can I have another 3 minutes? Is that okay?
The expense of questions.
Okay. No question is better, of course. What can I do? I never gave this talk, I never will. So, never mind. Okay. So, I want to talk about checkpoint therapy. This is where my lab works in immunotherapy. And I want to explain something. So, listen, be with me, guys. So, in the life of a lymphocyte, here is a slide. Lymphocytes, okay? Imagine you are a lymphocyte. So you grow up in the lymph node and you meet the nice dendritic cell. It presents some antigen, right? It can be a cancer tumor antigen. And you interact with it and you learn that this is the guy that you must be primed again. If you will see him again, you'll kill him, okay? Now you go into the tumor or in the plasma or whatever. If you meet the cancer guy, you kill him. Okay? Now, the story, as we learn the molecular biology, we actually learn that in addition to the interaction with the cancer, the antigen and the TCR that recognizes it, actually there are other interactions which excite or inhibit the activity of the T cell. And what we are learning now is actually to attack these interactions and via antibodies, monoclonal antibodies that we develop, we prohibit the inhibition of the T cells by the cancer cells. These are drugs, small molecules, okay? No need for expensive engineering cut. We just prevent what the cancer does to the T cells. Conceptually, it's simple. And there are various drugs, and just to show you, this is a clinical trial in melanoma. This is the first generation. They managed to achieve, you see, this is already for 10 years. No nonsense. So 20% of advanced melanoma patients, it was before, it was a death sentence. 20% are cured. And this is with a new drug, and this is with even a new drug. We now achieve 50% cure in advanced metastatic melanoma. Unbelievable. Combinations do wonders. The picture is complex because there are many different excitatory and inhibitory guys which we should learn. But just look at the potential. We are achieving, 50% cure now in some cancers just by attacking 2 of these inhibitory molecules. We are totally, completely the identification of all these guys. There is no reason why in the next very few years we can achieve much higher cures, unless I miss something by and large. And you can see now the rates of response achieving different types of cancer with checkpoint inhibition. The amount of drugs, the new drugs, is just exponentially increasing. Combinations, problems, I don't have time. Just to tell you that I've shown you these hallmarks of cancer. So hallmarks of cancer is composed of different processes on the molecular level. Each of these processes is now attacked by precision medicine and cancer immunotherapy. So it remains to be seen. Work is going on. And just a word on this cancer prevention. So, yes, so we will not prevent cancer today. And I'm prevented from talking about it, and I just wanted to thank David and wish him Mazal Tov and many happy returns.